All right, let's keep talking about neurons. We have a neuron right here. We've talked about neurons quite a bit already, but we will continue to do so because there's a lot to know about neurons. Now, one thing I want to repeat, and this is what we talked about in our last video, is that there is potassium leakage happening at the cell body, otherwise known as the soma. Soma and cell body are the same thing. And when a depolarization event is trying to spread across the cell body due to the influx of some cations, almost exclusively due to sodium influx, as the depolarization is spreading, that depolarization event is going to weaken due to the potassium leakage because there are potassium leakage channels within the cell membrane. Truth be known, there's certainly going to be sodium leakage channels as well, but sodium leakage channels are not going to detrimentally impact the graded potential. They're only going to aid the graded potential by adding more sodium into the system or into the cell body to help facilitate that depolarization event across the cell body or help the graded potential to reach the axon hillock. Once again, we want the graded potential to reach the axon hillock at a specific voltage. That voltage is negative 55 millivolts. Negative 55 millivolts represents threshold. If threshold is not met at the axon hillock, then we do not get an action potential down the axon. If we don't get that action potential traveling all the way down the axon, we do not get the release of neurotransmitters from the axon terminals. So, because there is potassium leakage, that graded potential needs to be large enough to compensate for any leakage of cations out of the cell. Once again, when cations leave a cell, they make a cell more negative or less positive. Now, one might ask, is potassium leakage an issue down the axon? And the answer is absolutely. There is going to be potassium leakage down axons, and that will impact the rate or velocity that the depolarization event of the action potential down the axon proceeds or propagates. The propagation down the axon of that depolarization event is certainly impacted by potassium leakage. Fortunately, our nervous system has ways to compensate for that, and that is due to what's known as myelination. Myelination is what I'm demonstrating here with these purple structures. In the central nervous system, myelination occurs due to the activity or a specialized cell known as oligodendrocytes. In the peripheral nervous system, it's due to Schwann cells. So in this example, it doesn't really matter to me if these are Schwann cells or oligodendrocytes, but what is important is the myelination, which is created by a lipid membrane wrapping around the axon, insulating the axon. And this insulation of the axon has two immediate impacts. One, anywhere there's a myelin sheath that we see in purple, there's going to be no potassium leakage. To be very clear, there's going to be no entry of ions into or out of the axon anywhere there's a myelin sheath. Between the myelin sheaths and these parts right here are what are referred to as the nodes of Ranvier. And the nodes of Ranvier are the only areas of the axon that are permeable to ions. And at the nodes of Ranvier, we have a litany of these voltage-gated sodium channels. The voltage-gated sodium channels open sequentially and propagate or move that depolarization event from the axon hillock all the way down to the axon terminal. But now these voltage-gated channels, that is to say, when the axon is myelinated, the voltage-gated channels are only at the nodes of Ranvier. And to be clear, there's probably going to be some potassium leakage channels at the nodes, but because these nodes are peppered with so many voltage-gated sodium channels, the potassium leakage is going to be negligible. So the nodes of Ranvier allow for increased propagation speed or velocity of the action potential, where it just jumps from one node to the next without having to go sequentially all the way down every piece of this axon until we get to the axon terminals. So One big function of myelination of axons is to increase the velocity or propagation speed of this action potential, or specifically the depolarization aspect of the action potential, or depolarization phase, from point A to point B, from the axon hillock to the axon terminals. This also is going to save energy for this cell. This cell is a neuron, it is a nerve cell, and anytime we have sodium moving into cells and potassium moving out of cells, we need something to maintain or restore those gradients, ensuring there's more potassium outside of cells and more, let me say that again, there's more sodium outside of cells and more potassium inside of cells. And as we've talked about previously in other videos, that is achieved or maintained by the sodium-potassium pump, otherwise known as the sodium-potassium ATPase. It is an enzyme. It breaks down ATP, liberating energy to pump ions against their gradients, sodium out of the cell and potassium back into the cell. We want to maintain those gradients, and the sodium-potassium pump achieves that, but it requires energy. The more voltage-gated sodium channels we have in this axon, the more sodium-potassium pumps we are going to need, and the more ATP we are going to need. So by having myelination along the axon via oligodendrocytes or Schwann cells, it is going to limit the amount of sodium-potassium pumps that we need. If there's less sodium-potassium pumps, which is the same thing as the sodium-potassium ATPase, if there are fewer of those, we are using less ATP, and we can save that ATP for other metabolic activities. One thing I didn't mention is that this propagation from the axon hillock to the axon terminals jumps from one node to the next rather than going sequentially through the axon. And that's what's known as saltatory conduction. If you know Spanish, one of the verbs to jump or to hop, just like that, is saltar. Saltatory conduction, to jump or to hop from one node to the next. And once again, that increases velocity of the action potential and saves ATP. Now, while we're talking about this, I keep mentioning graded potentials and action potentials. With regards to the neuron, the graded potential is confined to the cell body, and the action potential is along the axon down to the axon terminals. There are some differences between the graded potential and action potential. To be sure, there's commonalities. They both have a depolarization phase and they both have a repolarization phase. And for what it's worth, anytime a cell depolarizes, it certainly must repolarize before it depolarizes again. In this table right here, which is abbreviated, so I will go over this with you, this is highlighting some differences between graded potentials, GP, and action potentials, AP. One of them is the location. Graded potentials happen at the soma, otherwise known as the cell body, and certainly the dendrites. So we can say graded potentials happen at the cell body and dendrites, or soma and dendrites, whereas the action potential happens only along the axon down into the axon terminals. So location is certainly a distinct difference between the graded potential and the action potential. The second point we see right here is the electrical activity. What is it doing? A graded potential will depolarize a cell, so will an action potential. But a graded potential could also hyperpolarize the cell. That's bringing a cell below its normal resting state. If the normal resting state is, say, at negative 70 millivolts, it puts it down to negative 90 or even negative 100 millivolts. And the goal of that is to somewhat or entirely inhibit that neuron from firing. And we'll detail that more in a couple of minutes. So once again, a graded potential can be a depolarizing event or hyperpolarizing event, whereas an action potential can only be a depolarizing event. This third one is important. We've talked about action potentials being an all or none event or all or nothing event. That is to say, if this first voltage-gated sodium channel opens at the axon hillock, we can ensure that this is going to spread all the way down the axon. That's the all or none event. It happens or it does not. A graded potential is not an all or none event. it can be weak, and it can actually have multiple depolarization events. The graded potential is initiated by some sort of stimulus. It could be a ligand-gated channel, mechanically gated channel. It could be an aroma, some sort of pressure or change in voltage. But if repeated stimuli happen close enough together, those stimuli and depolarization events can be added up together to make a greater depolarization event. And the goal would be to make threshold here. So when we say a graded potential can be summed or summation, that's adding together multiple stimuli and multiple depolarization events that can happen as long as they are happening close enough together before the initial or previous depolarization events start repolarizing. Now, there's certainly some pathologies related to all of this with regards to the myelination. Multiple sclerosis is due to demyelination of axons, and that results in individuals having very, very, very slow action potentials down their axon to the axon terminals, and that has certainly some serious effects, musculoskeletal effects. So multiple sclerosis is due to the demyelination of axons. To be clear, not all axons have myelination. Some local anesthetics, now this is not a pathology, this is a medical practice, but some local anesthetics actually block these voltage-gated sodium channels. And by doing that, they don't get the release of transmitters, and it prevents any sort of sensation downstream. Called nociceptive neurons. These are neurons that elicit the feeling of pain. Pain is super important because it allows us to heal our tissues and prevent any more damage. And once again, these local anesthetics actually are going to block that and block the activity of these nociceptive neurons. Okay, let's talk a little bit more about electrical activity of cells and the changing of the electrical activity or voltage. Voltage is just the separation of charge between the inside and outside of the cell, any type of cell that has a voltage. And certainly neurons have a voltage in their excitable cells. That is to say, they can depolarize and repolarize. So I have a number of circles here that are representing cells, and we can envision these as being neurons because we're talking about neurons in the nervous system here. Just for space, I've drawn them as circles. Now I want to, once again, go over the normal action potential. I shouldn't say normal because nothing really is normal within the human body. It's somewhat variable. But we have an action potential. I'm having the resting state at negative 70 millivolts. Graded potential up to negative 55, which is threshold, depolarization event, repolarization event. Then we have hyperpolarization down here, and then back to the resting state. This cell right here is representing a normal cell. And this is the case with a muscle cell, a pancreatic beta cell, and a nerve cell, otherwise known as a neuron. What I want to point out here is that we've talked about sodium and potassium. Sodium and potassium are very influential in creating changes in electrical activity within cells. Within a resting cell is much more permeable to potassium than it is to sodium. And that's merely because there are more potassium channels in cells than sodium channels. And when I'm talking about channels here, I'm talking about leakage channels. So let me rephrase that. There's more potassium leakage channels than sodium leakage channels. So I'm showing here in orange, these are potassium leakage channels, and in purple, these are sodium leakage channels. And this is one reason the cell tends to have a negative voltage within it at its resting state, because there's more potassium leaving cells than sodium coming in. If there were equal amount of channels, sodium would actually have a bigger impact because it has an electrochemical gradient, whereas potassium only has that chemical gradient. But the reality is, because there's a lot more potassium channels, more potassium leaves resting cells than sodium coming in. And as a result, more positive charge leaves the cell than positive charge coming in, and it causes it to be slightly negative. Certainly, we can't forget about the negatively charged proteins within cells and the phosphate anions within cells. So this is our baseline. Now, there are certain metabolic conditions that can change this. And one of those conditions is known as hypokalemia. We'll talk about hyperkalemia in a moment, but hypokalemia is suggesting there's a diminished concentration of potassium ions in the extracellular fluid. So hypokalemia is not suggesting that there's no potassium in the extracellular fluid. It is just suggesting there's a lower concentration than normal. So if we look up here, I have drawn a very small potassium concentration here compared to what we see normally. And I put quotes around normally because what's normal? But this is the standard, let's say, and this is less potassium than normal. So hypokalemia is referring to the concentration of potassium outside of cells, outside of neurons. This could happen to diminished levels of potassium intake within the diet. It could certainly happen due to overhydration. Now, what impact does this have on an excitable cell like a neuron? Well, if there is a lower concentration of potassium out here, then more potassium is actually going to leave the cell via these potassium channels. More potassium is going to leave because there's a much larger chemical gradient. And the larger the chemical gradient, the steeper it is, the more movement of that specific ion. So in this case, in hypokalemia, more potassium is going to leave the cell than traditionally happens. If more potassium leaves, that is to say a positive charge is leaving the cell, what do you think is going to happen to the charge or voltage of the cell? If you said it becomes more negative, you're exactly right. As more positive charge leaves the cell down its steeper gradient because of the hypokalemia, it's going to make the interior of the cell more negative. So let's just make up a number and say that's negative 95. And if we go back to the graph right here, I am just going to put the resting state down here. This is a new resting membrane potential. RMP refers to resting membrane potential. And I'm not even writing negative 95 here. What I want you to see, what is important to see here, is that the new resting state is now further from threshold. So during states of hypokalemia, cells and neurons are going to require a much greater stimulus, a much higher valued graded potential to reach negative 55. So traditionally, the neuron would need to go from negative 70 millivolts to negative 55 millivolts. That's a change of only 15 millivolts. But if it's down to negative 95 millivolts, that's now a change of 35 millivolts, so you're going to need a much greater stimulus. So the problem with hypokalemia is neurons are not as reactive as they are normally. What about hyperkalemia? Hyperkalemia, we have a higher concentration of potassium ions in the extracellular fluid than normal. Normal, we have it looking like this. But what I'm trying to point out here is that there's higher than normal, so there's less of a gradient incentivizing potassium to leave cells. There is still more potassium in cells than outside of cells, but a less steep gradient. So potassium's not going to leave the cell as readily and as quickly and as much. As a result, more potassium is going to be retained in the cell. More cations are going to remain in the cell, more positive charges are going to remain in the cell, and it's going to make the cell, the resting state, more positive. So let's just say that's going to be negative 60 millivolts. If we're at negative 60 millivolts, the new resting state is going to be closer to threshold. And by being closer, this neuron is now going to be more reactive and become more excitable because it's much easier to reach threshold. Going from negative 60 millivolts to negative 55 is only going to require a change of 5 millivolts to reach that threshold and activate or initiate that action potential down the axon. So which is better for the body, hypokalemia or hyperkalemia? Neither of them. We don't want any of them, and that's why our body needs to be homeostatically regulated, regulating our levels or concentrations of potassium, our fluid volume, our water volume, and this also plays a part with other ions like sodium and calcium. Hypo and hypernatremia refer to the concentration changes of sodium in the extracellular fluid, and hypo and hypercalcemia refer to calcium. So we need to homeostatically regulate, our body needs to homeostatically regulate all of these variables to ensure things like our neurons are working normally. Now, there are times where our body, our nervous system is actually trying to induce similar reactions in the body. There are times where our brain is trying to mellow out neurons, tell the neurons don't fire as much. You don't need to be stimulated all the time. There is an amino acid known as GABA, gamma-aminobutyric acid, that opens up chloride channels in neurons. This is in the central nervous system. And by opening up chloride channels, what's going to happen? Well, to know what's going to happen, we need to understand chloride's gradients. I don't believe we've talked about chloride, but chloride has a much higher concentration outside of the cell than inside of the cell. To be clear, chloride is an anion. It's negatively charged chlorine, but chlorine is the atom. Chloride is a negatively charged atom or anion. There's a much higher concentration of chloride outside of cells than inside of cells. So when GABA opens up these chloride channels, the cell is going to be flooded with chloride. With this negative anion coming in, what do you think it's going to do? It's going to hyperpolarize the cell. More negative charge coming into the cell is going to make the resting state negative, further from threshold, and that neuron is not going to be as reactive. Glutamate and aspartate are also amino acids that function as neurotransmitters. So we'll talk in a subsequent video on a number of different neurotransmitters that neurons release. But one form of neurotransmitters are amino acids. Amino acids are building blocks of proteins, but they also do other things in the body, one of which is they serve as neurotransmitters. So GABA, glutamate, and aspartate are amino acids, but they're also neurotransmitters. Glutamate and aspartate are excitatory neurotransmitters, whereas GABA is an inhibitory neurotransmitter. We say it's inhibitory because it brings that resting state further from threshold, and it's going to require a much larger stimulus. Glutamate and aspartate, on the other hand, are excitatory neurotransmitters within the central nervous system, and they move the resting state closer to threshold. So what I've been talking about with GABA, glutamate, and aspartate, the physiological response is similar to hypokalemia and hyperkalemia, but hypokalemia and hyperkalemia is pathological, whereas these are intended activities to induce similar results. And as far as GABA goes, a lot of anesthesia that are given via gas for surgeries stimulate GABA to hyperpolarized cells, putting them further from threshold so that patient doesn't feel anything, they don't have any level of consciousness, they're not feeling any pain. Okay, so let's take another look at the electrical activity of a neuron. And this is specific to the action potential that runs along the axon, and specifically that is activated by those voltage-gated sodium channels. Once again, at the axon hillock, if threshold is met at negative 55 millivolts, the action potential or depolarization event is initiated, and it propagates that action potential down the axon. So here's the action potential in blue. The only value I have here is negative 55, which is representing threshold. So we have a depolarization event, a repolarization event, hyperpolarization. I don't want to suggest that hyperpolarization always occurs, but it often occurs. And then hyperpolarization back to the resting state. We have two phases in here known as that are very important. One is referred to as the absolute refractory period and the relative refractory period. Refractory, I believe in Latin, means stubborn. And that is suggesting the neuron is being somewhat stubborn or reticent to engage in another depolarization event or another action potential. During this period right here where the neuron begins to depolarize until it's fully repolarized right here, that purple dot is the point at which the cell or the neuron is fully repolarized and it's back to its resting state. During this temporal period, keep in mind this x-axis is time, during this period is the absolute refractory period. It is the point or phase where we cannot have another action potential. We cannot have another depolarization event. And I'll talk about why in a moment. The relative refractory period is we can have another depolarization event, another action potential, but it's going to require a much larger stimulus. And the relative refractory period might be easier to understand. That is to say, during the relative refractory period, the neuron is in the hyperpolarized state. So let's just say right here, we're at roughly, let's say, negative 90 millivolts. The normal resting state is at negative 70. If the cell is hyperpolarized, one, it is fully repolarized, so we don't have to worry about waiting for it to repolarize. But it is this undershoot or hyperpolarization means it's going to require a much larger stimulus, a much larger graded potential to get back up to threshold. So this dashed line right here is representing threshold. So during the relative refractory period, a neuron can have another action potential, but it's going to require a much larger graded potential to make or to reach threshold at the axon hillock. Now back to the absolute refractory period. Why can we not have any electrical activity or specifically a depolarization event or the initiation of another action potential during this time? Well, one easy answer is because the cell has not fully repolarized. A cell generally, specifically a neuron, needs to fully repolarize before it depolarizes again. That's not always the case. It's certainly not the case with skeletal muscle. But with a neuron, this absolute refractory period coincides with full repolarization of the cell. But if we want to look at the mechanical activity that actually influences this, we need to look at these voltage-gated sodium channels. These are all the same cell or neuron. These would be along the axon. This is a voltage-gated sodium channel in all of these cases right here. And these channels, which is really one channel, it's just 3 different phases, have 2 gates. The red gate at the top is the activation gate, and the red gate at the bottom is the inactivation gate. So at rest, the activation gate is closed, and the inactivation gate is open. In this case, sodium cannot enter the cell because that gate is not open. So when the channel is closed, sodium can't move into the cell. Keep in mind these are voltage-gated channels. When there's a change in voltage, that is to say when this graded potential reaches threshold, it's going to open up that channel. We're going to get sodium influx. So instead of the channel opening and closing really fast, what happens, if we can see this, I don't know that we can see this, in activation gate here, activation gate here, this opens and that closes. So it probably happens like that. And even though that's almost happening simultaneously, it's allowing enough time for sufficient amount of sodium to come in to depolarize the cell, to activate those other voltage-gated sodium channels, to propagate that impulse downstream to the axon terminals.